All right, hello everybody. Welcome to the HIRM seminar for today. It's our pleasure to have Olivia Rissland as our seminar speaker today. Dr. Rissland obtained her Bachelor's of Science with honors in mathematics and Latin at Brown University. And then she went on to do her PhD at Oxford University as a Rhodes Scholar and worked on RNA uridylation as a novel mechanism of gene regulation. Of course, that's a very hot topic at the moment. And then went on to do her postdoctoral fellowship in Dave Bartel's lab at the Whitehead. Following that, she started her independent career as an assistant professor at the University of Toronto. I believe it was in the Hospital for Sick Children, and then moved to the University of Colorado School of Medicine, where she's currently assistant professor in the Department of Biochemistry and Molecular Genetics. So she's a real expert in RNA protein interactions and RNA metabolism, and it's wonderful to have her with us today. So Olivia, the stage is yours. You could share your screen.
Great. All right. Well, thank you so much. Let me just get this screen sharing started. Need to do the obligatory. Everything looks okay to everyone else. We're all good. Okay.
That's great. Thanks.
Well, it's a real pleasure to be here. And it's a real pleasure to see so many faces that I haven't seen in a while, thanks to COVID. And today what I'm going to be telling you about is really revolves on a project in the lab thinking about how RNA binding proteins themselves are controlled, and thinking about this especially in the context of embryogenesis. So before I get into the science, I really need to acknowledge the people in my lab that have really driven this project. The work I'm going to be telling you about was started by Michael in the lab, then taken over by Svetlana and Lauren, and currently Jesslyn is doing some of the newer unpublished work that I'll be showing you. So I have had a long fascination with how gene expression is controlled. And so, of course, this is not a new idea to any of you, but when we think about the sequence of DNA, it has a ton of information. It, of course, contains information about what should be expressed, but also how much, when, and where. And as we all know, these last three questions really fall under the umbrella of the control of gene expression. Now, gene expression is fundamentally important. It matters for all organisms. This could be prokaryotes, yeast, my cat when he was a kitten, my favorite Red Sox player. I am, in fact, from the Boston area, so I'm very excited to put this up for other Boston area people, David Ortiz. And I think, the real question for us as scientists is how do we go from what the sequence of DNA is to understanding about what this means for gene regulation? And of course, the control of gene regulation is complicated because on the one hand, gene expression has to be reproducible in terms of when we think about processes like development, but it also has to be able to respond to changes in the internal and external environment. And so, of course, the way that we have evolved to deal with this is by having multiple levels of gene regulation. So when we think about the life cycle of an RNA molecule, we have control in the nucleus with processing, export through prime end formation. And then, of course, we have events in the cytoplasm, which have really fascinated me since I started graduate school. And predominantly, we're thinking about translation and mRNA decay, although to be fully honest, today's talk is actually going to verge into another area of gene regulation, which is protein degradation. But the real point that I want to make is that all of these layers of regulation is really what allows the cells to be robust and also be responsive. So in my lab, historically, we've thought quite a lot about how translation and mRNA decay talk to one another. How are these processes mechanistically related? And how do these processes change during development? And so sort of broadly speaking, we put these into these two umbrellas of these mechanistic types of projects, and then really asking questions about how the framework of mRNA decay and translation talk to each other during development. And one thing that is not on here, but one thing that we are increasingly thinking about is not just protein production and mRNA decay, but also protein destruction and mRNA decay. And how do the two different ways that we can degrade biomolecules, how do these processes also talk to one another? And this sort of new idea in the lab has really been inspired by the work that I'm going to be talking about today, which started from a question of how is translation, how is mRNA decay, how is that framework controlled itself during development? So I put this slide in really for the trainees, which is to say that we have been a little intentional in terms of how we approach science. And to be honest, this was really inspired quite a lot by Dave, my postdoctoral mentor, in terms of thinking about how do you begin to answer a question? If you have a biological question, how do you choose which system or which tools to go about? And so we really have taken the approach that we don't want to be tied just to one model system. We really want to use the best model system that we can to answer the question that we're asking. And this, I think, has meant that we really have three fundamental model systems in the lab, and both of which have their strengths and their weaknesses. So for instance, cell culture is obviously really great for mechanistic studies. It's going to be pretty bad for a developmental study. And on the other hand, we have Drosophila, a great model system, a little bit harder to use for mechanistic studies. And we also have an area in the lab that I'm not going to be talking about today, but has really centered around the protozoa Giardia lamblia. This is actually what causes beaver fever, the disease that you get when you drink unfiltered water, up in the White Mountains. We are really interested in Giardia because it also has a lot of streamlined molecular machinery, and it's very, very distantly related to traditional model systems. And so this system allows us to ask a lot of questions about how these fundamental molecular machines have evolved in eukaryotes. So today, what we're really going to zoom in on is we're going to zoom in on Drosophila, and we're really going to be thinking about gene regulation in early embryogenesis, and particularly during the maternal to zygotic transition. Now, this is a key step in animal development. It really happens in all animals. And the fundamental is that the developing oocyte is loaded up with maternal gene products. And because the initial stages of embryogenesis don't have transcription, those initial events are actually driven by the maternal gene products. And so what will happen during development is that you lose the maternal gene products and you start making zygotic ones. And so then by the end of the maternal to zygotic transition, or the MZT as we call it, by the end of that, you're now under the control of zygotic gene products and the zygotic genome. So this has been a really classic system for RNA biologists, and it's been studied quite a lot for really two main reasons. The first is that it's a place where we have a lot of regulated mRNA decay and a lot of post-transcriptional gene regulation, especially early on where there's no transcription. So post-transcriptional gene regulation plays this outside role. And then we also have this activation of transcription that occurs and starts producing zygotic transcripts. And so this has been obviously hugely interesting for RNA biologists. What I want you to take home from this is that there are really two key fundamental things that have to happen during the maternal to zygotic transition. The first is that we need to start initiating zygotic transcription. And the second is that we'll need to start clearing maternal mRNAs. Now, we're going to be talking about this in Drosophila. And so I need to introduce another player to you that's going to come up, which is something called the Pangu kinase. So in most animals, the MZT is set off by fertilization. In Drosophila, it's a little bit different. And the main thing that sets off the MZT is something called egg activation. And the way this works essentially is that the mechanical pressure as the oocyte passes through the oviduct, this activates the egg. This leads to the activation of the Pangu kinase. Then the Pangu kinase really serves as this master regulator to set off all of these events in the MZT. And so if we zoom in on this a little bit, what the Pangu kinase does is, it does a lot of things. It phosphorylates a lot of proteins. It leads to changes in translational activation and repression. And some of the consequences of that is that we start getting translation of transcription factors, which will enable zygotic genome activation. And we start getting translation of mRNA decay factors, which will then enable maternal mRNA clearance. Now, so far, we've really been thinking about the RNA aspect of this. And perhaps not surprisingly, given the fact that I have loved RNA since graduate school. But really, the question that we're going to be wrestling with today is, what about the proteome? What happens to the maternal proteins that are loaded into the oocyte? Are they actively degraded, or do they just sort of hang about in the embryo? And where this question really started from were some, just to be honest, some very serendipitous, unplanned experiments that led us to start thinking very seriously about three RNA binding proteins. So the ones that we're really going to be focusing on today is a helicase called ME31B. This is known as DHH1 in yeast, also DDX6 in mammals. Another protein called trailer hitch, and then a final protein called CUP. Not going to get into sort of the long backstory of why we started thinking about this, but we knew already as we were beginning our experiments that these proteins are really, really important for oogenesis, and they're really central RNA binding proteins. So they form a complex together that represses gene expression. In the early embryo and in the oocyte, this complex really appears to repress translation. The way that this does so is that CUP actually interacts with EIF4E, and it does so at the very place where EIF4G would bind EIF4E. So essentially what that does is it blocks the formation of a true initiation complex. All three proteins have, as I mentioned, have been implicated in translational repression, but they're also very, very involved in mRNA decay. These are a conserved set of interactions. You see them really throughout eukaryotes, but including Xenopus, Plasmodium, C. elegans. And for the Drosophil biologists, these have really been very important because they're implicated in repressing key genes during oogenesis and embryogenesis. So when these popped up on our radars, we knew that from a developmental context, they were very important, sort of as RNA binding proteins. They were very interesting to us. And so we started asking a really fundamental question, which is, okay, well, what are their levels like during the MCT? And this was carried out by two trainees that were in the lab actually back in Toronto, Miranda and Michael, and they just did a very simple experiment, which was to harvest lysates, stage lysates during embryogenesis. You essentially collect lysates at hour intervals, and then we just ran them out on a gel and did a couple of Western blots for proteins that we thought might be interesting. So what I'm showing you here are 6 Western blots, and we're probing for 6 different proteins. And so on the left, what you see are proteins like EIF4G, poly-A binding protein EIF4E. And what I've marked with the red arrow is about when zygotic genome activation happens in Drosophil. It's about 2 to 3 hours. And to also orient you, by the end of 5 hours, we really have done the real bulk of the MCT, and we're kind of beginning to exit from the MCT part of embryogenesis. So I think you'll all agree with me that there's really nothing much to write home about for EIF4G, poly-A binding protein, EIF4E. But we saw something very, very different when we looked at these 3 proteins that we were really fascinated by: CUP, trailer hitch, and ME31B. And what we noticed straight away was that their levels really dramatically decreased right around the same time point as zygotic genome activation. And I should say, actually, that work from Elmer Valle has shown that the levels of these proteins in the early embryo, they're actually incredibly highly expressed. So ME31B is thought to be at concentrations of 15 micromolar. So in fact, although its levels decrease sort of around 20-fold at this 2 to 3 hour time point, it's actually still quite abundant afterwards, but you still just get this massive decrease. And this was a really interesting observation for us. And it was made even more interesting because when we looked in the literature at other organisms, what we saw was that the orthologs of ME31B decrease at essentially equivalent developmental time points. So always right around zygotic genome activation in C. elegans or Xenopus, you'll see that the levels of the ME31B version also decrease. And so that made us think there must be something important going on here. So we really wanted to understand why this happened. And so the first thing that we thought about was, well, Pangu is this real master regulator of all things MCT. Is Pangu required for the loss of ME31B, CUP, and trailer hitch? And so to answer this question, Miranda and Michael, again, isolated lysates from embryos, staged. We did some Western blots, and I think what you can see very clearly is that in the absence of Pangu activity, where we have a mutation in one of the Pangu subunits, you see complete stabilization of the proteins. So this was all well and good. We sort of knew at this point that we had these proteins that we knew from the literature. They repress key genes during oogenesis and embryogenesis. It actually turns out that they don't just repress key genes, they repress thousands of transcripts in the early embryo. We knew that they were rapidly destroyed during the MZT. This required the Pengu kinase. And we had this really intriguing result from Terry or Weaver, the Whitehead, that all three proteins were also phosphorylated by Pengu, which was a very intriguing observation to us. And so in the long term, we'd really like to know why these proteins are degraded. I have to say, as a sort of a full spoiler, we can't actually quite yet answer the question of why it's important that they are destroyed. But we thought that was quite a heavy lift. And so we thought we would start with at least a how. How are these proteins destroyed? What mediates the destruction of ME31B cup and trailer hitch? And really, we were hoping to get to a place where we could connect our genetic observation of the requirement of Pengu with sort of a more molecular mechanistic explanation for it. So at this point, to be honest, I wasn't really trained in Drosophila. And so we hadn't really made use of the awesome power of Drosophila genetics, but we thought that this might be a good time to dip our feet into those waters. And so what we thought about doing was we could use sort of a medium scale shRNA screen to identify the genes that were required for ME31B degradation. And essentially the way this would work is that we had a Drosophila line that expressed ME31B GFP from the endogenous locus. So the ME31B GFP behaved almost identically to the wild type copy. And the idea was pretty easy. You take embryos, live image them, they would start off green, they would become less green as the MZT proceeds. And of course, if you knock down a gene that's required for it, the embryos would still be green. And that would be a way that we could at least begin to get some more players involved in this. So before going down this route, the first thing that Michael wanted to do was he wanted to ask the question of like, does the scheme actually work? Could we actually use microscopy as opposed to Western blots for visualizing the degradation of ME31B? So what I'm showing you here are just some representative images of live imaging of embryos. And this essentially is a wild type embryos. And what we've done is we have false colored the fluorescence. And so what this means is that the more fluorescence they are, the greener the embryos are, the hotter the colors are going to be that I'm showing you. And then as the fluorescence goes away, the embryos get cooler. And so you can see exactly what we saw in Western blots, which is that the embryos start off pretty hot and then they get cooler as you go through development. And sort of very reassuringly, exactly as we predicted, if you now take an embryo, essentially the same set of embryos for the Western blots that I showed you earlier, you can now see that the hotness is maintained throughout the time course. Again, this subunit is required. So the first question we wanted to do was, well, the Pengu kinase actually has 3 different subunits. Previously, we had only interrogated the role of one of the subunits. And so we wanted to ask the question of, well, if we knock down the other 2 subunits, does that also stabilize ME31B? Is the full kinase required for ME31B degradation? So one thing I should also say is that because of the types of experiments that we're doing, we're really thinking about protein decay, and we had expected that we would eventually go down the road of thinking about E2s and E3s. We didn't want to knock down these components in the whole fly. We thought that might actually be quite problematic for the flies in some cases, and could lead to a lot of confounding phenotypes. And so what we did was we actually drove expression of the shRNA during oogenesis. So we knocked down the gene of interest during oogenesis, and then we're looking for phenotypes in the embryo. So what I'm about to show you is that we're going to knock down individual components of the Pengu kinase during oogenesis, and then again, look at what happens to ME31B during embryogenesis. So consistent with the allele results I showed you on the previous slide, if we knock down one of the subunits GNU, or actually the other subunit plutonium, again, we see this complete stabilization of ME31B. And to be honest, this was actually quite reassuring that the whole entire scheme sort of worked, and it gave us a lot of confidence moving forward to doing this medium scale screen. This also really confirmed for us that really was the entire Pengu kinase that was required, which was consistent with our previous results, and then led us to ask the question of whether or not the degradation was triggered by egg activation or fertilization. As I said, in Drosophila, these are 2 real independent triggers that will happen. We suspected that fertilization would not be required because the Pengu kinase really acts downstream of egg activation. We wanted to test this directly. So what I'm showing you here are time course images from fertilized embryos versus unfertilized embryos. And really in contrast, what you see when you remove the Pangu kinase, the embryos don't really care about fertilization. Really in all cases, sort of get almost wild type dynamics. And so this really confirmed for us that as the egg activation through the Pangu kinase, that was what was leading to ME31B degradation. So this was great. I mean, this was something, let me put it that way, but it really wasn't where we were trying to go. What we were really trying to get to was a mechanistic understanding. And so this meant that we had to begin to think much more seriously about protein decay, which, to be honest, was not something that I had really thought about since my undergraduate time. So for those of you who also have not thought about protein decay in a while, just to remind you of how this happens, is that some of the majority of protein decay is mediated through the ubiquitin proteasome system. And so what happens is that a target protein is ubiquitinated, it's polyubiquitin, you get these polyubiquitin chains, and this really acts as a tag for the cellular garbage disposal of the proteasome to target those proteins. So what's really key in controlling protein decay is the addition of these ubiquitin chains, and that is going to be determined by what's called the E3 ligase. So the E3 ligase recognizes the target, and then the E3 itself is recognized by an E2 ubiquitin activating enzyme, and then there's an E1 that lies upstream of that. So you want to understand protein degradation, you really want to understand what is the E3 that is targeting the protein, and you might also want to understand the E2 as well. So in Drosophila, we have several hundred E3s. There are about, I think, 40 E2s, and then there's one E1. And so we were really asking in the long run, what is the E2 and E3? And the first place we want to start off with, because there are other systems that degrade proteins, is it really the ubiquitin proteasome system? Is this where we should be thinking? So the first thing we did was we actually asked a related question, which was, is autophagy, which is another pathway of degrading proteins, is this required for ME31B degradation? And so we did this by, again, knocking down various autophagy components in oogenesis, looking at ME31B degradation. And I'm just showing you two representative knockdowns. What you can see is essentially there is really no effect. There's no stabilization of ME31B. And so this made us feel pretty confident that ME31B is not degraded by autophagy. The next thing we looked at was whether we could see evidence of ubiquitination of ME31B. There are several different ways you can look at this, but one real classic way is to pull down your protein of interest in very stringent conditions. So essentially you remove associated proteins, you're really only looking at this protein, and then do a Western blot looking for ubiquitin in your IPs. And so what we're looking at here, first are lysates. That top gel is probing for ubiquitin. You see lots of ubiquitinated proteins. When we pull down ME31B GFP using this GFP antibody, what you can see is we can see the ME31B GFP in the pull down, and we can also see this ubiquitinated smear, which is really quite strong evidence that ME31B is in fact ubiquitinated. And one thing that was quite nice was when we repeated this in Pangu lysates, in essentially when you have Pangu mutants, we lost this ubiquitination. And this is very consistent with the conclusion that Pangu activity is required for ME31B degradation. So then the final thing we want to know is, did the degradation of ME31B require the proteasome? Now, I have to say, with full honesty, that this actually turned out to be a slightly trickier experiment than I had imagined, and perhaps I had pitched to Michael when I suggested we do this. And this really has to do with the fact that if you really strongly inhibit the proteasome, the flies don't lay eggs. And if they don't lay eggs, it's very hard to look at embryogenesis. And so the majority of our knockdowns, in fact, we got no eggs. So what I'm showing you here is microscopy from one of the knockdowns, which did lay eggs. We suspect that this is probably only a partial inhibition of proteasome activity, just because of the other phenotypes that we saw. But even in this scenario, we can see a stabilization of ME31B, and I'm showing this quantitation of the fluorescence on the right. In a contemporaneous study from Howard Lipschitz and Elmar Valle, they actually injected embryos with a proteasome inhibitor, and they also saw stabilization of ME31B. And so I think the evidence is pretty strong that the proteasome was involved. So the question that we're really building up to is, okay, well, what are the enzymes responsible for the ubiquitination? Really, to be honest, what is the E3 ligase? So to answer this question, Michael in the lab screened about 100 E2 and E3 components, and some E3s are multi-subunits, so he would knock down essentially as many E3s as he could get RNAi lines for. This turned out to be a bit of a slog. He definitely screened RNAi line after RNAi line after RNAi line, and I think for about 9 months, we didn't really get anything. We had really expected that you would knock down some component and you would see this complete stabilization the way we had with Pengu. And he was getting really to the end, and we were beginning to think like, well, maybe we're wrong. Maybe it's not what we think it is. And I think it ended up being his second to last batch of RNAi lines that he was trying when he came to my office. And I should say that Michael was pretty stoic as a scientist. He doesn't really have highs. He didn't really have lows. He just sort of kind of did his fly work. But he came to my office and he was really excited. And I was like, okay, I think there's something good going on, because he doesn't get excited. And he's like, I think we got a hit. Okay, I want to see the microscopy. Show me the data. And so what he showed me was a knockdown of an E2. And this was very exciting to us because it was really the first time where we saw a microscopy that looked so reminiscent of what we saw with the Pengu kinase mutants, which is you really saw this very robust stabilization of ME31B. So the next thing that we wanted to do was we wanted to, because we're looking at GFP fluorescence, we wanted to confirm first that this happened for the endogenous ME31B, but also for cup and trailer hitch, which I haven't really mentioned at all since those original Western plots. So again, Michael, I got staged lysates from control and UBC-E2H knockdown lines, probed for ME31B. He also probed for cup and trailer hitch. And what you can see is that it's not just ME31B GFP, but it's also the endogenous ME31B and cup and trailer hitch, all of which are stabilized almost completely in this UBC-E2H knockdown. Now, of course, we were in flies, so we had to take an advantage of this to come up with a really exciting name for our E2. And Laura White, who's a graduate student in the lab next to ours, reminded us that at this point was really where there was that great Netflix show going on about Marie Kondo and the KonMari method, which is a method of simplifying and organizing your home to get rid of objects that don't spark joy. And so we thought this would be a really great name for an E2. And so we renamed UBC-E2H as Marie Kondo, or Kato, as I'll be calling it for the rest of the talk. So this was great. This was our first real handle on the system, but we had a bit of a puzzle still, which is that we did not have an E3 ligase. And we knew there really had to be one. And so we were still left with this fundamental question of what is the E3 ligase? And so this is where I think we lucked out a bit, to be honest. So it turns out that Kato is actually fairly well conserved. So there are orthologs in yeast and humans, and actually in both the yeast and humans, that those E2 orthologs interact with the same complex. In yeast, it's called the GID complex. In humans, it's called the CTLH complex, and it's a multi-subunit E3 ligase. And based on homology, we could identify essentially the homologous proteins in Drosophila. But what was really striking to us was we also had some IP mass spec data where we had pulled down ME31B, or we had pulled down CUP to look for interacting proteins. And what we noticed was that 5 of these 6 subunits interacted with ME31B and interacted with CUP. And so this suggested that there was not just that we had the E2 implicated, but that we also had a physical interaction with the E2, with the E3 associated with that E2. And so the really interesting thing for us that made us feel even more positive about this is that actually when we were doing these experiments, these genes in flybase, where we had essentially picked our initial E3s, they weren't annotated as E3s. And so when we were choosing that original list of RNAi lines to order, we hadn't picked any of these because they weren't E3s. And so in fact, in our initial screen, we hadn't tried any of these. And so that really made us feel really interested. Is the CTLH complex the missing E3 ligase? So I'm just going to show you one representative image where we knock down one of these subunits. Here I'm showing you RAN BPM. And what you see is that essentially a phenotype that's very, very similar to what you see with Pengu or Cato knockdowns, which is that you see this real stabilization of ME31B GFP. And in fact, when we do Western blots, we see something very similar for endogenous ME31B and cup and trailer hitch. And so this really led us to feel very confident in saying that, yes, the CTLH complex is in fact the E3 ligase responsible for degrading ME31B, trailer hitch, and cup. And so, of course, we had to continue and name some of these CG names. The CG7611, although it is implicated in the human complex, we actually didn't see it in the Drosophila embryonic ones. It's either not involved in the embryonic complex or it's not present. We don't really know. And we called the other ones, with some help from Yuki Tamari, various Japanese words for cleaning, broom, and tidying up. And I'm going to coming back to some of these a little bit later on in my talk. So we still had a little bit of a mystery, which is we knew the players involved now. So at least we had that, but we really didn't have a link to how Pengu activity stimulated ME31B decay. And as I mentioned early on, some really beautiful work from Terry Orr-Weaver's lab had suggested that not just ME31B, but also cup and trailer hitch were all phosphorylated by Pengu. And I should say that Pengu actually phosphorylates many, many, many RNA binding proteins, which is very interesting in and of itself. But our initial hypothesis was that this phosphorylation acted as a phosphodegron for the CTLH complex. So essentially, in oocytes, ME31B, for instance, is not phosphorylated. There's no recognition by the CTLH complex. As you go through egg activation, you have Pengu activity, you phosphorylate ME31B, and now that stimulates recognition of the phosphorylated ME31B by the CTLH complex, thusly leading to its degradation. So we decided to test this prediction in a few different ways. And I should say, as a side note, one of the most obvious ways would be to make phosphomutants of ME31B such that it couldn't be phosphorylated. One challenge of this is that ME31B, cup, and trailer hitch are all phosphorylated in many places. And so we felt that that was not going to be a really great strategy of looking at this. So the first prediction, if this model were true, is that the CTLH complex should not interact with ME31B in the absence of Pengu activity, that this phosphorylation, if it really is making a phosphodegron, should be required for recognition by the CTLH complex. So to look at this, we decided to use immunoprecipitations followed by Western blots. So this is an example from wild type embryos. We're pulling down with GFP, so we're pulling down ME31B GFP, and we're looking for one of the CTLH subunits called RAN BPM. And what you can see is that in the pull downs, but on the control IPs, we see this interaction between ME31B and RAN BPM. So the real question is, well, what happens in Pengu mutants? And so a real big surprise was that we could still detect the interactions. And this was really our first line of evidence that this phosphodegron model might have been overly simplistic and, to be honest, probably wrong. The other prediction of the model is that the CTLH complex should not interact with ME31B in oocytes. So in oocytes, they've not yet gone through egg activation. The Pengu kinase hasn't been activated yet, or all of that should be sort of before any of those events occur. And so you'd expect, if this phosphodegron model is really correct, or if it's really important, if this interaction is stimulated by egg activation, that we would not see it in oocytes. And so Jesselyn in the lab actually did this quite recently, where again, she IP'd ME31B GFP from stage 14 oocytes. Now we're probing for a different member of the complex called muscalin, but again, you can detect an interaction between ME31B and a CTLH subunit. And so really taken together, this led us away from the idea that there was this phosphodegron and that the way that Pengu kinase activity stimulated the degradation was via this phosphorylation idea. So again, how does the Pengu kinase stimulate the destruction of ME31B? So to answer this, we were actually very lucky that a really beautiful paper from Dave came out, I guess, almost 5 years ago at this point, where Stephen in the lab had done ribosome profiling during oogenesis and into embryogenesis. And so we remembered that one thing that the Pengu kinase really does is it leads to the translational activation of many mRNAs, but also the translational repression. And so we thought, well, let's look at the CTLH subunits. Let's look at CATO. Let's ask how their translation changes during this egg activation time point. And we really didn't see anything to write home about for CTLH subunits, but what really jumped out at us was that CATO translational efficiency dramatically increases as you go from stage 14 oocytes to 0 to 1 hour embryos. This is right when egg activation occurs. And so in fact, based on ribosome profiling, its translation increases probably 30, 35 fold. And that actually puts it in like the top 10 of all mRNAs in the transcriptome that are translationally activated. So, was this the link with the Pengu kinase? Can this translational activation explain how the Pengu kinase works? In that same paper from Dave, they also did ribosome profiling in Pengu mutant stage 14 oocytes and activated eggs. And what I'm plotting for you here is the translational efficiency of CATO now in these Pengu mutants. And what you see is there's no activation. And so what this is really telling us is that somehow Pengu activity leads to this massive translational upregulation of CATO. And we can see this when we look by Western blots using an antibody that we made against CATO, is that in wild type embryos, you start off, you get this really big production of CATO protein, which we don't see in the Pengu mutant embryos. And so what this all means is that Pengu activity, by changing translation, is actually then able to sculpt transcription by production of transcription factors. It's able to shape mRNA decay by translation and production of mRNA decay factors. And it's actually also able to shape protein degradation during the MZT through at least the production of CATO. So the question that recently we have really been struggling with is the fact that ME31B is actually pretty ubiquitously expressed, as is the CTLH complex and as is CATO. And so really the main question that we've been asking is, well, why is this degradation seemingly so specific to the early embryo? So if you look in RNA-seq, as I said, ME31B, CATO, CTLH complex, they're very widely expressed. They're expressed in cell lines like S2 cells, they're expressed in the adult fly, they're sort of expressed everywhere. But in general, ME31B is actually stable. And so that, to be honest, was how we got into this whole entire thing in the first place, was this very unusual instability of ME31B during the MZT. And so this leads to a really interesting question of what mediates this developmental specificity? Where does that come from? And so in the last few slides of my talk, I'm going to show you, it's all unpublished essentially data, where we're really going to start thinking about how protein decay of this like key RNA binding protein, how we can have developmental specificity around it. So to get into this question, we're going to need to dive in a little bit more into the CTLH complex. And we were very lucky in thinking about this question that last year, a really beautiful set of cryo-EM structures came out from Brenda Schulman's lab, looking at the Serravissier-GID complex. So this essentially is the Serravissier version of the CTLH complex. And what was really nice about it was it's a complex that has 5 to 6 members in it, subunits in it. They're sort of organized in different ways and they play different roles. And her structure really helped organize where these were and what their interaction interfaces were. So in the complex, we have this catalytic module, we have a scaffold module that helps connect the catalytic module up with a substrate receptor module. And so the first thing that we did when we were really beginning to think much more deeply about the regulation of ME31B degradation was where the Drosophila versions proteins fit in. So we were able to quite easily identify the catalytic proteins involved in the Drosophila CTLH complex. We could identify RAN-BPM and HUKI as being part of this scaffold module. And we'll say that muscalin is still a little mysterious as to where it is in the complex. It's not actually in the yeast version. But one thing that really jumped out at us and that was very intriguing was we could not identify any subunits that would be involved in substrate recognition. So the really key players in Serravissier, GID4 and GID5 that really recognizes the substrate, they don't really seem to exist in Drosophila. And even more intriguingly, when we threaded the Drosophila versions through the yeast structure, although broadly they were the same, when we zoom in at these interfaces between the Drosophila GID8 and this GID5 that we can't see, in the yeast version, GID8 has this tail sort of wraps around GID5 and helps anchor it in place. In the Drosophila version, the Drosophila GID8, which is hooky, there's none of that tail. There's nothing to wrap around. This really makes us think that how the Drosophila CTLH complex recognizes substrates is really fundamentally different than what you see in yeast, and actually, as it turns out, what you see in humans, which is really fascinating. And so we have really begun to think that to understand how the degradation of ME31B is controlled, we actually also really need to understand how it's recognized as a substrate, that these two questions are really inextricably interlinked. So one clue that came out for us was when we started trying to IP the CTLH complex using knocking down various members of the complex. And I'm not going to take you through all the data. I'm just going to show you what we think to be the most intriguing observation. So in this experiment, we're pulling down ME31B GFP using a GFP antibody, and we're probing for the CTLH complex with RAN BPM. What really jumped out at us was when we knocked down muscarine, this subunit I told you we didn't really know what to do with. When we knocked down muscarine, we lost the interaction between ME31B and RAN BPM. And this really had made us wonder whether muscarine might be the substrate adapter. So to test this a little bit more fully, what we did was we changed things around. So now we're going to IP muscarine. We're going to look for the other different subunits in ME31B, and we're going to do this in the presence or absence of RAN BPM. And so what I'm showing you here are control knockdowns. Again, we're IPing muscarine, and we can detect interactions with other components of the CTLH complex, RAN BPM, Katazuki, and ME31B. Now, what we think is really compelling evidence is that when we knock down muscarine, we lose the interaction with Katazuki, which is part of the catalytic module. Sorry, when we knock down RAN BPM, we lose that interaction between muscarine and the catalytic module, but we maintain the interaction between muscarine and ME31B. And so what we're beginning to think is that muscarine is acting as a substrate adapter. And we think this is really critical in thinking about how the degradation of ME31B is restricted, because muscarine is only transcribed in oogenesis. And so of all the CTLH subunits, muscarine is the one subunit that shows tissue specificity that coincides sort of right with where we begin to see ME31B degradation. So this is good. This is good. This is beginning to tell us, how we could get this tissue specificity. But one thing that we haven't really talked about is what happens at the end of the MCT. Why doesn't the CTLH complex just keep on degrading ME31B? Why doesn't it keep on degrading trailer hitch? Why doesn't that continue beyond the MCT? And so this is what we're really going to end with, which is thinking about why and how this protein degradation of ME31B stops. And so what we noticed, again, going back to staged lysates, is that it turns out that both the CTLH complex, both subunits within the CTLH complex and CATO itself are actually also destroyed at the end of the MCT. So here are some Western blots from staged lysates. We're using PAB as a loading control. You can see, as we have seen many times in the past, ME31B GFP goes away. But what's really interesting is if you look at CATO, you see it being produced, and then you see it going away. And what's actually really fascinating is that you also see this for two CTLH subunits, muscarine and hooky. So both these also go away at the MCT. And so this, to me, is so intriguing, because what we're beginning to see is not just that ME31B is an example, sort of like the one example of a maternally deposited protein being degraded, but muscarine and hooky are also maternally deposited, and they're also going away, but they're doing so with different kinetics than ME31B. And so this really suggests to me that thinking about protein degradation during the MCT that this is a much richer set of gene regulations than perhaps we had previously thought about. The last thing that I really want to show you is some new data we got just like last week that I'm just so excited. I wanted to share it with you all today, which is it's known in the protein decay field that oftentimes E2s and E3s can actually self-inactivate. And so we had this slightly cockamamie experiment of looking at musclin levels and Hookie levels in Cato knockout embryos. So, obviously ME31B is not degraded in these embryos as we've seen before, but what's really interesting is that now musclin and Hookie are also not degraded. And so this raises this really intriguing possibility, not just do we have more examples of maternal proteins being degraded, but that they are part of some feedback loop where perhaps once ME31B cup and trailer hitch are removed, then the E3 itself begins to self-inactivate. So really what we're beginning to think is that in terms of thinking about protein degradation during the MCT and thinking really specifically about ME31B as sort of a model for how to approach this, is that there is a really rich set of regulatory steps that occur here that are acting at the transcriptional level, they're acting at the translational level, and they're actually acting at the protein decay level in terms of how this activity of the CTLH complex for ME31B is developmentally restricted. So what I've told you is that we have this active CTLH complex that really acts on maternally deposited ME31B. We think that musclin is a substrate adapter protein there. We need to do a lot more work to really validate that hypothesis. And the really key thing here is that prior to egg activation, there's no degradation of ME31B. So in fact, the CTLH complex seems to still be able to recognize ME31B, but it's poised and not active because the E2 has not yet been made. Cato has not yet been made, and it won't be made until egg activation occurs. So we go from a poised CTLH complex to an active CTLH complex. And then finally, this activity itself is shut off at the end of the MZT by degradation of two, actually three, I should have updated my slide, which is Hookie, Cato, and musclin are all seem to be also degraded at the end of the MZT. And so this really closes the end of the chapter on ME31B degradation. And then in the rest of the fly, we know these proteins come back, they begin to be expressed, but what we suspect happens is there's a different substrate adapter that is expressed. It's not musclin because musclin transcription is restricted to the oocyte. And so now we have a whole different set of proteins that are degraded by the CTLH complex. And so we think this is a really, really fascinating. We're really interested in, again, how protein and mRNA decay pathways talk to one another. And to be honest, even though we'd like to think of ourselves as an RNA lab, we are really interested in protein degradation during the MCT. Did we happen to sort of luck into this really cool, fascinating pathway? Or are there many other types of proteins that are degraded? Are there other RNA binding proteins whose degradation is controlled during the MCT? And what might those pathways be? So with that, I think I have hopefully left enough time for questions. I really need to acknowledge everybody in the lab who did all of these experiments. The two Michaels who were in the lab, Miranda, Lawrence, Svetlana, were all just amazing and spearheading these projects to begin with. Jesselyn has now sort of picked up the project and is adding her own twist to it, which is great. I want to thank our funding agencies. And I'd also like to say for the trainees in the audience, if you're looking for a postdoc and you're excited about Colorado, we have lots of mountains. If this is interesting to you, please send me an email. I'd love to talk more about that. So with that, I will stop. Thank you for your attention, and I'm happy to take any questions.
Great. Well, thank you very much, Olivia. That was fantastic. So if there are questions, please feel free to write them into the chat or unmute yourselves and ask your questions. Maybe while people are thinking and typing, I might ask the first question. So this musclin sounds fascinating, and you said that it's not found in yeast. How about in zebrafish or xenopus, which have well described maternal to zygotic transitions, right?
Yeah, I am 98% certain it's found in zebrafish and xenophis. It is in humans, which makes me feel that's where I sort of get that level of confidence from. We have not looked at it in zebrafish or in xenophis. We don't really know anything about it there. I will say the one thing that's very interesting in general about the CTLH complex is um, this is a very different set of substrate proteins from what has been described in yeast, where it really seems to be much more involved in glucose metabolism, and in humans, actually, where it's been much more suggested to be involved in DNA damage. And so there does seem to be some plasticity involved in its targets. And so I'm not sure a priori whether, muscarine might be involved in doing something similar to what we see in Drosophila and xenophis or zebrafish, or for instance, another E3 could sort of be taking the place of this E3.
Mhm. Great. Any other questions? Just unmute yourselves or type into the chat.
Olivia, hi, it's Alan. Do you know about the mammalian cereblon ubiquitin ligases?
Um,
I'll
I'm
send
sorry.
you a link. I'll send you
Okay.
a link to a paper, but it's a similar story, a small molecule regulated E3 ligase that only with a certain small molecule present will you get E3 ligase activity. And I could imagine that it might be of future interest. So I'll send you a link to that.
Yeah, no, that is definitely, that would definitely be great. I will say, one thing we have this model that it's sort of just muscarine that could be controlling this. I will say that we have also entertained alternative hypotheses that there might be other things that help restrict this developmentally. So one thing we've been trying to do, so in S2 cells, which are a very common Drosophila line, cell line, they don't express muscarine. And so we've been trying to add back muscarine to ask if we can get ME31B degradation. We have not yet gotten those experiments to work, we think, for technical reasons, but there is this small voice in the back of my head that's like, well, maybe there's something else going on that we don't yet fully understand about this interaction, and that something like a small molecule or another post-translational modification of muscarine or ME31B or something else is also an important component of the story that we don't yet understand. So that would be great. Thank you. I feel like I saw something in the chat, but I have no idea where the chat now went on my screen.
Olivia, maybe this is in your papers, but have you made the lysine to arginine substitutions? Have you been able to map the lysines that are important on ME31B?
We have not in our paper. In Howard's paper, they did. They were able to identify some. We have thought about this. So there's like a couple areas of hesitation that I've had with doing that experiment. And one has to do with one puzzle to us is cup, trailer hitch, and ME31B are part of this complex. They all sort of behave the same, as like we can't seem to ever separate them from each other, if that makes sense. They all just sort of behave identically. And so we're still not really sure in a way what is recognized, which of the ubiquitinations is really key. And so I think I've been hesitant with doing those types of experiments because I'm not sure what we would do with if we didn't see a difference, is there an alternative hypothesis that has to do with this slight complexity that we have? So my inclination has been, if we could recapitulate this in an S2 system, then we would have a much better handle to do those types of experiments. I'll also say the other wrinkle is that ME31B, all these proteins are really important for oogenesis. So you could also run into you disrupt its function too much in other ways, and then you've just messed up oogenesis in a way that's pretty uninterpretable for these types of experiments.
Thanks.
Yeah.
So maybe while other people are thinking, I'll ask again. So you mentioned this complex's involvement in DNA damage response, and I'm wondering whether there's a translational angle here where potentially this complex could be involved in disease. And I'm just wondering, have you noticed any connection there? Are these factors disease genes, particularly sort of cancer genes? I would imagine would be high on that list.
Yeah, that is a great question. I will say, I want to say the mouse version of RAM-BPM is actually linked with infertility, which has been a very intriguing observation to us. We have not thought about this too much in the context of cancer, and I think that would be a really interesting direction to go in. But I have nothing really intelligent to say about that.
No worries. Yeah.
Yeah.
It sounds intriguing though. All right. Are there any last minute questions? If not, let's thank Olivia again. It was a fantastic talk. Really appreciate you joining us today. And thank everybody for attending, and we'll see you at our next event, which I believe is next week.
Thank you so much, everyone. It was a pleasure.
I think Olivia, if you want to hang on, but no, I think he's gone. All right. We'll see you later. Thanks, everybody. Bye.